Assalamu alaikum, dear students. It is good to see you again. Before starting your new lesson, I should thank you for the following of your lessons. And before starting your new lesson, you should have a flashback at your previous video and lesson. If you remember, we studied about the cell theory. For now, you understand the definition of the cell, that it is the fundamental structural and functional unit of all living beings that possess humans, animals and plants. As you see your own body, you may understand that your body is made up of different type of cells, structures, and they carry out different type of functions. So the question arises this, that how it was possible to understand these different type of cells and their structures and functions. Today's lesson will talk about these things. In this video, you will be able to follow these objectives. First, the microscope and the history of microscope, types of microscope, differences between electron microscope and light microscope. So let's start about the microscope itself. At first, you should understand what does the word microscope means. It is from ancient Greek words. Micros means a small and scopian means to look or see. And simply we can say that a microscope is an instrument which facilitates the observer to see tiny things into large scales. So about the history of microscope, we can say that it is an old history. 1, 2, 3. About history of the microscope, we can say that it is an old history. In order to understand the history of the cell, you should have a flashback at the early days of 13th century, when the first guy whose name was Sullivan de Ahmad invented eyeglass. It was quite simple but useful. So he started the invention method of microscope. After him, the Zachariah Johnson and his father Hans, in 1519, they invented the first optical telescope or compound microscopes. This is the first compound microscope which relates to these two persons. It was about the history of the microscope when the invention of the microscope started, and for today's types of the microscope, we understand that we possess more than 10 types of microscope, but mainly we will use two microscopes in school. The first light microscope, the second electron microscope. So in this video, we will talk about these two important microscopes. One light microscope, another electron microscopes. But these are the other microscopes that they are possessing their own function and importance in biological fields. So let's talk about two important microscopes. First, the light microscope, and it is a referring name, light microscope. It uses visible light and a system of lenses to generate magnified image of small objects. If you see the light microscope at picture, you may understand that it possesses different parts. The first part that you put to your eye is connected to the objective lens. That is called eyepiece, and this is called objective lens. And this is the body that you will hold this microscope. This is the stage where a specimen or the sample is put to see, and these two are focusing systems. This is the base, and this is the light source, which is connected to this machinery to see tiny and small parts into large scales. Let's talk about another important microscope, especially the electron microscopes. As you know, it is also a referring name, electron microscope, which means they will use a beam of accelerated electron as a source of illumination. This is the invention of two guys and scientists in 1931 by the physicist Ernest Rosca, this one, and the electrical engineer Max Null, this one. They both work together to invent the electron microscope. In order to understand the function of electron microscope, you should understand the specific differences between electron microscope and light microscope. One of the most important differences between the light microscope and electron microscope is this, that they use different type of sources for illumination. Light microscopes are using photons that they possess 200 nanometers, but the electron microscope are using electrons. Why? This is the reason. Because the wavelength of an electron can be up to 100,000 times shorter than that of visible light photons. So to understand the function of the electron microscope, we can see the video example. Before starting the video, I will show you how electrons are produced, how it will pass through different parts of electron microscope, and how it will reach the specimen or the sample that we put on the stage. So let's begin it. At the first stage, the electrons are going to be produced by electron gun. Then a beam of electron will carry through a vertical path, passing from different type of magnetic lenses and also scanning coils, and after passing the backstart electron detector, it will reach the sample. After reaching the sample, it will eject from the sample. Then it is going to be counted by secondary electron detectors or backscattered electrons, and at the final stage it will produce the final image. So let's start and see the video example. This is an electron microscope. At the top you see that electron is going to be produced by electric gun. Then it will pass from the anodes. If you know electrons are possessing negative charge, so it is easy to lead it toward the sample that is located on the stage. So the beam passes through a pair of scanning coil and a pair of deflector plates in the electron column that deflect the beam in X and Y axis. As you see, when the electrons reach at the sample, then after ejecting the electrons, this is the X-ray detector, backscatter detector, and also the gas secondary detector. They will combine these electrical or electron signals in order to produce the final image at a screen, which could be like your television. Thank you so much, dear students. See you next time on next video. If you possess or have any kind of difficulties and questions, you may inform us. Thank you so much. Goodbye.